microbiology, proteomics, cancer biology, molecular biology, nanotechnology, bioremediation studies, and animal studies. He has trained thousands of students and faculty during his PhD. Currently, he has handled 200 plus projects from 2024 at his firm, and he has given 50 plus students placements. He have 10 publications and one book chapter. So I would like to introduce Mr. Ramesh. Please take over to this and continue the session.
Hello? Hello? Hello?
So you're audible.
Yeah, am I audible?
Yes.
Just a minute. Hello? 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 Can you hear me?
Yes,
Ashmita,
sir, you're audible.
ma'am, I need
Yes.
access to share my screen.
Yes, I will give it to you.
Yeah, yeah. Sorry, I got a call from a guide so that. Hello? Okay, sir. Just Excuse me, ma'am. I have joined with two, like one is with laptop. Maybe I got access to my mobile. Can you give it to another K Ramesh so that I can share? Hello?
Hello.
Madam, can you give me no access?
Yes, sir. Please join with your another account so that we can give the access. We are not able to see another K Ramesh.
Okay, only one is visible. Just a minute.
Yes, yes. Only one is there.
Just a minute. I'll. Yeah, maybe I'm trying with mobile only. Just a minute.
Hello.
Sir,
Hello.
good.
Is there any problem in screen sharing?
Yes, sir.
So try to solve it, Prathamesh.
Yes.
Yes. You are. It is visible now.
I can see this logo also, maybe.
Yes. So you can start, Ramesh, sir. Ramesh,
Yes, sir.
and totally
I
36
think,
participants are there, audience.
okay, sir.
Asmita ma'am is there.
Yes.
Hello, Asmita.
Yes, sir, I'm there.
Okay, so I'm little late. I will introduce.
Yeah.
So you can start, Ramesh.
Yes, thank you, sir. Thank you, sir. Thank you for the opportunity. Yeah, good evening, everyone. Just a minute. Yeah, good evening, everyone. So I'm Dr. Ramesh from Osman University, Hyderabad. So today I'm presenting a lecture on cell culture techniques and its applications. So before that, I think so I think there are around 36 participants. So how many of you are aware of cell culture techniques? Can I get any messages? Just rapid response.
No, very few, very few.
Okay, very
So
few.
you
Okay,
are
sir.
sure
Okay, sir.
that no one knows about cell
Okay.
culture technique.
Okay, sir. So to make you understand, just I'll compare with the bacterial culture, like how we culture the bacteria, so that it will be easy for you to understand. So I'll go with the introduction. So cell culture is nothing but as we grow bacteria in the petri dishes, tissues, and different culture media. So here we can grow any part of an organism, tissue or cells can be grown in vitro condition. So you can see in vitro cultivation of organs, tissues, and cells at confined environment or outside the body by providing suitable environment is nothing but cell culture technology, or it is also known as tissue culture. So here the we can grow like any cell, a part of organism, like it may be animal or plant cell in a controlled environments by providing all physiological our conditions in outside the body. So this was like extensively used in all fields of life sciences nowadays, like maybe a, biotechnologist, microbiologist. Not only that, any plant people with the extract they were checking for anti-cancer activity, like any drugs, like we can see we can study the behavior of the cells and developing new drugs, production of vaccine, all nutrient compound, like any cosmetics also to test it. We cannot do it directly onto the animals. So before going into the animal levels, so we can everything tested on the cell culture. So the cell culture like includes normal cell line as well as cancer cell lines. So if you want to test for any cytotoxicity, like you can see any cells, like you can see there are different types of cells can be grown in.
Hello.
Yes, madam.
Hello. Sir,
Hello.
good evening, sir. Good evening, sir. Sir, could
Yeah.
you please remove that logo from your slide, because it takes most of the part of the. I'm seeing in the mobile, so majority of your slide is covered with that
Yeah.
logo.
Yes.
Sir.
Yes. Maybe it's clear now.
Thank you so
Yeah,
much.
it's clear now, right? Yeah.
Thank you
Sorry.
so much.
Sorry. Yeah, it's clear, ma'am. Now I think it's clear.
So much.
Yeah. So we can grow different types of cells, any cells, like we can grow like previously we tried like chick embryo we will use now, like Kama says regularly. We can take that embryo, we can grow fibroblast cell lines, whatever we want. That is called primary cell line. There are different cultures of cell line. Here we can see the basic, like we can remove tissue from any organism and we can chop or we can just mince it and we can digest with different enzyme, and we can place it in a cell culture plates by providing medium, so that can be grown simply. This is the basic of cell culture, you can see. So coming to some historical developments in cell culture, you can see Claude Bernard, he proposed the physiological system of an organism can be maintained in living system. So even that after death of an organism, like we can grow some part of the body in the in vitro conditions. And you can see some other scientists like Rouse, here coming to 1907, Harrison, he was the first person cultivated frog nerve cells in a lymph gland, and he observed the growth of nerve fibers in vitro for several weeks. So he was considered the father of cell culture. And further developments maybe Carroll. So he introduced aseptic techniques. So cells could be cultured for a long period. And then Gay in 1952, he established a continuous cell line from a human cervical carcinoma, which is famously known as HeLa. That is from Hela Henrietta. She is a woman. She was suffered with the cervical cancer. Though this was the first established cell line. And then Eagle studied nutrient requirement of selected cells in a culture, and he established chemically defined medium. Like we use different media for growth of bacteria. So even the cells can be grown in different media. So we'll use different media. So we'll be discussing in next slides. So like then Sato established serum-free media from cocktails of hormones and growth factors. Then the human insulin was the first recombinant protein to be licensed as a therapeutic agent by cell culture, and human growth hormones was produced from recombinant bacteria was active for therapeutic uses. So these are some historical events. So coming to the major development cell culture, we know that growing a cell or any tissue from an organism living system outside the host when we were maintaining. So we have to maintain a lot of things like humidity, temperature, and other things, and it should be like maintained sterile conditions has to be maintained. Sorry. Okay. So the first development of cell culture is was the use of antibiotics. See, the cell culture medium, it consists of lot of highly nutritious things like amino acids, proteins, salts, and everything. So it is more potent, or it is highly potent of risk contamination. So use of antibiotics which inhibit the growth of contaminants, the first development in the cell culture. Then the second was the use of trypsin, because there are cells, like the cells can be two types. The cells are attached adherent. Another type is suspension, like we know blood cells, which continually floating in our body. Such cells are called suspension cell lines. And the cells which are attached to the substrate or the bottom is known as adherent cells. So while growing the cells, we have to take them out. We have to detach from the surface. So earlier there was like some issues with the detachment. So the second invention was in use of trypsin to remove the adherent cells from the subculture. Furthermore, from the culture vessel, this was another development. And third one was the using of chemically defined culture medium. Earlier, different scientists tried with different medias, blood and other protein agents to grow cells here. But this development, major development like antibiotics, trypsin, and culture media. So after these developments, so the cell culture, like it gone like major developments was happened. So here coming to this, so what is the cell culture used for? Why we are discussing here? So there are like technologies currently playing major role in anywhere, like in all fields of life sciences. Like you can see model system for studying basic cell biology, interaction between the disease causing agents and cells, and even you can see the effects of drug. We cannot test it directly onto the humans or any living system. So before which we can use it, like if you want to test some cosmetic onto like any organism, before which you can grow some skin cell line. So you can give this any drug or the cosmetic onto that cell line. We can test it how it is interacting with the skin cells. Like such types we can be studied in the cell lines, and the process recurring of aging, nutritional studies, and everything can be tested in the cell lines. And also it can be used for testing toxicity. Like you can see a lot of plant extract or any chemical compounds which were synthesized. No, so to know the toxicity or cytotoxicity of the compounds and drugs, any extract can be studied on cell lines. On various cell lines you can be tested, and it is mostly useful in cancer research. That is the various chemicals, like even the virus, radiation, other things can be studied in the cell culture. And it can be used in even virology for cultivation of viruses for production of vaccines, and also used to study the infectious cycles of different organisms, viruses. And it can also be used in genetic engineering, like production of commercial protein and viruses for use in vaccine production. There are different vaccines which are produced by using this technology. And gene therapy also, we were using cell culture. I think you might know about CAR T cell therapy. They will take out the patient's blood out, and the chimeric antigen receptor can be modified by using genetic engineering, and the cells can be grown in the tissue culture, cell culture, and the cells will target the cancer. When target maybe it may be cancerous or any tumors can be targeted. So it is advancing, and also using disease modeling. So for studying cancer, genetic diseases, and infectious disease, and metabolic disease and gene functions can be studied using this cell culture technology. So before getting into this, so I just want to tell you, like, as you are new into this this cell culture, so some terms will be used in cell culture so that you will understand better. Maybe we'll use confluency. So generally, the cell culture, the cells will be grown in a tissue, like tea flask will be. I will show you next one. The cells can be grown in different sizes of tea flask, which is started from 12.5 mm to it's 140, 240 like that. There are different sizes are available depending on the requirement availability. So like when we are growing cells in a flask, so when the flask is completely, the air, whatever the bottom is there, that 25 centimeter, if we are growing a T25 flask, if that area is completely covered by the cells, that confluence we can, how much of area? Sorry. Sorry. Yeah. Sorry. So how much of area is covered by the cells? I mean the cell growth. So if that area 25 centimeter area completely covered by the cell growth, that is called 100% confluency. So depending on the size, how much they covered, 50%, 60%, 80 and 100%. So the word we use, the term we use called confluency. So that for doing any experiment or anything we were going into cell culture, so minimum 80% of confluence is required. So the cell has to reach 80% confluence, then only we can do further, or we can use it further the cells. Then the splitting is nothing but, as we do know, subculturing for bacterial culture. So here also with the cells, once the cell is completely, I mean, confluent, 100% confluent. So when you want to grow more cells, for example, you need 100 million cells for some experiment. So the T25 flask can grow up to 2 to 3 million cells. So you need 10 million cells. So for that, you need to grow them more and more. For that, when the flask is completely filled, so you have to.
Excuse me, sir.
Yes.
Screen at not share. Your side.
Oh. Just I can see. Okay.
दादी से पूछ कर आओ, दादी को बोलो बाहर आने को, जाओ.
The participants, please mute yourself.
Hello. Hello.
Yes, sir. Yes.
Ashmita ma'am, I just shared to you, shared my PP to you. Can you just scroll it now? Better, I think, with the host, it is better, I think. I
Yes,
just
just
shared
a
it on
minute.
WhatsApp. Hello.
You can share your presentation with the moderator otherwise. Dr. Ramesh, can you hear me?
Yes, yes, yes.
Why don't you give your PPTs in PDF format or something?
Yeah. I just share it actually. Just a minute.
निकलोगे बिल में से अंदर बाहर.
Sharing
हाँ
from
मम्मी,
your
बिल
mobile.
में घुसे लोगे.
It is not very-
Yeah, I got it. Just a minute.
धक्का लग गई थी. हाँ. हम चाहे हैं फिर ऐसे करिए. ऐसे किया उसने.
Yeah. Yeah. Did you get the term confluency?
आपको बुलाने नहीं आएगा चाय ठंडा.
Somebody has mic.
Yeah. Can you just switch it off now?
मैं अब किसी को बुलाने नहीं भेजूंगी, भाभी को भी नहीं बोलूंगी जाने।
Somebody has to mute the other participants. Yeah, you can start. You can go ahead.
Yeah. Did you get the term concurrency, like the area which is covered by the cell growth? Okay. The next term is splitting, like as we subculture our bacterial culture, like that, so we can subculture it, like we can grow it, like from when the cells grown completely in T25 flask, so we can split into two flasks or 4 flasks according to the growth of the cells, depending on that. And another one is seeding. The term seeding, like for just separating the cells into different flasks, like 6 well, 12 well, 96 well, depending on the type of experiment. So when you seed the cells after counting, so because here we use a specific number of cells for each experiment. For example, to testing for cytotoxicity, we'll see 10 to 15,000 cells in a well. So for before that, we have to count the number of cells, how many cells are there, how many cells you require. So first we have to grow them, how much you need. Then after we have to seed them, like with the number of cells should be uniform in all 96 well plate. Then only you can study your drug or compound properly. So that is seeding. And here we use media, like the media, there are two types of media we can use. The plain media is nothing but the medium without ABS. Generally we use ABS, that is nothing but fetal bovine serum. So that for growth of replication of the cells. When sometimes we use plain medium without ABS, such medium is called plain medium. When we add FBS, that is 10% to plain media, that is there are different medias like MEM, DMEM, Delbecqu's modified Eagle medium, and modified Eagle's medium. These are the commonly used medias in cell culture. When we add FBS to this plain media, that will become complete media. So media plus FBS is called complete media. And serum starvation, you can see the term. So for example, I'm doing some cytotoxicity testing. So I have seeded cells in 96 well plate. So then they have to reach 80% confidence, and then I cannot add my compounds directly. So before that, the cells may be in different cell cycle stages. Like some cells will be in G1, G2, S phase and different phases. So to take all the cells into common cell cycle phase, we have to starve the cells. Like we have to take out the complete medium from the wells, and we'll add only plain medium without ABS, so that the cells will not replicate. So the whatever the cell cycle stages they are in, so they all come to the normal stage at the same stage. So then only we can give the treatment, so that your compound testing will be very efficient. Okay. And another term is called trypsinization. So as we just the second development in cell culture, use of trypsin. So as the cells, most of the cells are adherent, the cells are attached to the bottom. So to take out the cells, so we'll add trypsin. That is around 0.25% of trypsin will be used. So when we to remove the cells from the flask or the attached area, so that term is called trypsinization. Counting, you know that to count the same number of cells. So generally we use chemocytometer to count the cells. Then freezing, this is term, I think after our experiment, mostly like we'll get cells from different places. So we cannot buy the cells always, or we can get the
Hello,
The mic is off.
is there any problem?
You are mute. Ramesh, you are mute.
Ramesh, you are mute. Please unmute yourself, Ramesh. He is not mute, but some problem is there. I think internet problem is there.
He was mute, sir, in between.
No, now he is not mute.
No, you see there are, okay, yes. Very useful presentation otherwise.
Yeah, excellent. His presentation is very good.
And very informative also for the student perspective. Those who
Yes.
are actually the cell culture work.
Correct. Yes. Those
Because
who,
bacteria are the one which contaminates actually the cell culture experiment.
yes.
And we have to struggle days together.
Correct. Very correct. So the precautious hearing this lecture
Exactly. Unless
is
or
a
otherwise
very,
we know
very
where these problems are, we will not be able to address those issues.
correct. Ramesh, you can call me on personal phone or you can chat in the write in the chat box. Ramesh, if you have any problem, you write in the chat box, please. Your presentation is very good. So, in the meantime, he will be ready. In the meantime, I know Prakash Halami, sir, but
Yes, sir.
Dr. Praval is there. Dr. Praval, you are from? Dr. Purnima, Dr. Amruta, please answer, please. Dr. Tofel, means all these doctors, they are PhD, but they are listening to the lecture. So
Exactly.
can you please?
Or if any participant has any basic questions, we
Yeah,
can.
I know Arif. Arif is there. But
Now Ramesh is back, so
Yeah, sir. Sorry, there is
oh,
something,
okay, you proceed. Ramesh,
yeah,
proceed.
yeah, sir. Okay, sir.
Yes, go ahead, Ramesh.
Okay, sir. Just a minute. Again, I'm not getting the screen sharing option, sir. I share it to Ashmita, madam. Can you share it, madam?
Yes, you can send to me. I will.
Just
Prathamesh,
okay. Better, sir. I'll
Prathamesh,
just a minute. Okay, sir. Just a minute.
you can send to me. We'll
Yes, sir.
share and you continue
Yeah,
without
sir.
sharing.
Yeah, better. Yeah. Just check your WhatsApp, sir. Just.
Okay.
Yeah.
Okay. You can proceed.
Yeah. Just I need just share it, sir, so that yeah. So I think I don't know where it is disconnected. Can you just maybe the terminology I'm using, different terminology I'm just explaining you. Okay. There are like confluency, splitting, cryopreservation, revival. There are different terms will be used. Yeah. So the cell culture, like types of cell culture, you can see there are based on the source and growth characteristics, the cell culture are different types, the cells are different types. You can see by source or duration, the cells like primary cell culture and secondary cell culture. You can see the primary cell culture is nothing but where the cells can isolate it directly from the normal tissue. You can see like any liver, any tissue like liver, any tumor, or any kidney, any organ, or where you can get it directly. When you grow it in vitro, that is called primary cell culture. So they'll be having the parent tissues characteristics. They have limited lifespan. Like we cannot continue them. Like we cannot grow them for more passages. Yeah, I forgot to mention one more. Can you hear me? Is it clear?
Yes.
Yeah.
Yes, sir.
Only
So one.
presentation we are
Sir,
unable
your
to see.
screen are not shown.
Yes.
Exactly.
Deshmukh sir is just sharing the screen. Just a minute. I think. Sir, we were waiting for this. Yeah. Before that, you can ask me like if you want anything before sir sharing the screen.
Yeah, you can tell your practical experience with relation to the cell culture work.
Yeah. So like I was into this last 6 to 8 years. I was into cell culture under my PI, Dr. Sandipta Burgula at Department of Microbiology, Osman University. We'll be doing screening for different compounds. Like we were helping physics and chemistry people after their, compound screen or after they synthesize the compounds. We'll be testing for anti-cancer properties. So we'll be doing regularly cytotoxicity testing, like
What
Yeah.
cell culture, what cell line you use for cancer cell line?
So currently we have cervical cancer like HeLa, SIHA, breast
Okay.
cancer MCF7 and MDMB231, and even we have like some normal cell line like HCK, like human embryonic kidney cell line, HCK293, and even we have some K569, which is a leukemia blood cancer cell, and we have HepG2 liver cell, and lung cancer cell lines also we have, and even we have some macrophages like raw macrophages also we are having all these cell lines. So depends on the requirement. We'll be doing some collaboration works at my PI's lab. Now at my company we are establishing this facility. We got almost like maybe within next by end of maybe the next 15 days we are going to establish completely. So we'll be doing all these assays starting from screening. Like if you give any compound, so that compound can be screened for whether it has any anti-cancer properties or not, starting from MTT, which is called cell viability assay or MTT assay, cytotoxicity assay, and we'll be doing different assays called scratch assay and clonogenic inhibition assays and CAM assays will be doing, like to confirm whether your compound has any anti-cancer properties. So this final assay we'll be doing is CAM assay. So where we use 9 to 11 days embryonated eggs will induce cancer by using the selected cancer cell line, and along with that we'll be injecting our compound drug, whatever the drug you are testing. So generally the cancer cells, they produce a lot of blood vessels, that is called angiogenesis, to grab the nutrients from different nearby areas. So like if your compound having this anti-cancer properties, that should inhibit the formation of angiogenesis, that we will be testing. So like in 11 days old egg, so will induce cancer, then along with that we'll inject our drug. After 72 hours of incubation, we'll harvest the this egg, and we'll take out the cam layer and we'll count the number of blood vessels in it. So we'll be having placebo without any treatment, and we'll be using anti-cancer drug when the positive control, like which is available in the market, and we'll be testing our compounds along with that. Yeah. Yeah. Can you rotate the screen, sir? Yeah. Yeah. So there are different assays, sir. So according to the requirement, like we'll be doing in collaboration, whatever they ask, any type of cancer studies we'll be doing so far, starting from
The
screening.
cell line is from the animal or from fish?
Mostly human cell lines will be using, and it depends. Like whatever we have, mostly human, sir. Human cancer, normal cell lines will be.
So this picture is only for a pictorial.
Yeah, this is to just show you how the cells can be grown. Like this is like primary cell lines. Like we can take it from any living organism. We can take a tissue of it. We can just crush it, and we can just place it in a cell culture medium. The cells can be grown simply.
Very good.
Yeah. So previously we did with chick embryo fibroblast. Like we got a 7-day-old chick embryo. So we got it and we just harvested it, and we got all the internal organs out, and whatever the tissue we got it, we just digested by using trypsin, and that can be just macerated, and we just placed it in the cell culture medium. We grown it. Even now this is 4th year, still we are able to revive the cells. We stored them. We use it for some antiviral activity, for some others collaborative work. So like that we can use any cells can be grown actually. So depends on the requirement. Yeah. Can you move to next sir? Next slide. Hello. Yeah. Next. So these are just developments of cell culture from like starting. Yeah. Next, sir. Next.
1 minute, because
Yes.
again I have lost it. Again, I'm regaining. I'm
Yeah.
trying
Okay,
to
sir.
regain.
Okay, sir. Just got it. Yeah, these three are the major advancements in cell culture technology. You can see use of antibiotics. Mostly
Correct.
we use penicillin and
So
streptomycin.
you tell me the slide.
Yeah. So like these are the developments. Like earlier, they used to get a lot of contaminations in the cell culture, and they are like they struggle to take out the cells from the bottom, because most of the cell lines are they are attached to the bottom. And this was like overcome by using this use of antibiotics. We use penicillin and streptomycin, which is antibiotic solution in 1% will be using to avoid any contaminants in the cell culture medium. So after
Whether the slides are visible? Slides are visible?
Yes, sir, we can see the slides now.
yeah.
So you can guide me.
Contamination problem is there even now also.
Yes, sir. It's
We
there
use
definitely,
one.
but we have to be very careful. If you do it, like if any single person is maintaining the cell culture,
Yeah,
it's okay.
exactly.
When different people, multiple people entering into cell culture, like so we have to be very careful because use this media is highly nutritious, no sir. That is the one reason. So we use FBS. It is very nutritious. So that's what there are a lot of more chances of getting contamination. So we have to be very careful while working with the cell lines. We have to like wear all the like gloves and everything. We have to be, and only we allow only 2 people maximum into cell culture while working. So when you maintain all these things, then only we can maintain the cell culture without any contamination. Otherwise, repeatedly we will be getting contamination, and we have to waste a lot of medias. The cell culture flask, even they are more costly. One box costing around 20,000, 15 to 20,000 of each box. So that we have to be very careful. And the trypsin, like we use, like there are different types of cells actually. What is the other end which is attached to the bottom? I told you. So when you add trypsin, within 1 or 2 minutes, the cells can be detached. And another one is cell culture medium. So like we have different medias for growth of bacteria. So even for cell culture, there are different medias like MEM, which is minimal essential media, DMEM, Delbecque's modified Eagle medium, and RPM, that is Roswell Park Memorial Institute. So these are the 3 common medias we use regularly, like Newton agar and whatever we use in our bacterial culturing. So these are the different media. So earlier there was no defined media, but now we have chemically defined media. Are a lot of media. There are different media are available other than the 3 medias. So there are so many medias. So like depending on the cell type requirement, we can use different medias in cell culture. Next slide, sir. Yeah, you can see. So the cell culture technology, like different areas will be using model system for studying cell biology, interaction between disease causing agents and cells, and the effects of drug on cells, process, triggering aging, nutritional studies, how toxicity testing as we discussed, like for different components, we can test for toxicity, whether they are cytotoxic or not. And so you can study the effects of new drugs and compounds, and for studying of various chemicals, viruses, radiation, other things can be also used. Maybe the genetic engineering, it's like virology to produce vaccines. So we can cultivate any viruses, like as I said, no, like we established this chick embryo fibroblast is a primary cell line we established to check some compound against antiviral properties. So we used duck plague virus. So she has some compounds, it's a phytochemical actually. Yeah, I think there is a message from Prerna Deshmukh. So no autoclave are used in cell culture. You know, madam. So because we cannot autoclave the cell culture medium, as it has highly nutrient like proteins, other things, so that may be coagulated. So here what we do is we we use autoclaved water when we are preparing media manually. So there are 2 options. One is we can get powder medium, you can prepare cell culture media. Another one is we can get ready-made media available nowadays. So the powder medium, once you get it, so we have to use double distilled sterile water, and we'll be preparing everything inside the flow, laminar flow biosafety cabinet. So we'll add this antibiotic solution after adding all these things. Then after will be sterilized by using filter sterilization. We use where we use 0.2 micron syringe filters. We cannot autoclave the cell culture medium. So we'll use filter sterilization only. Okay. So next slide, sir. Yeah. So production of commercial proteins and vaccines, there are different things can be produced by using cell culture. And the cell culture can be used for gene therapy. Maybe, you know, like CAR T cell therapy also one of it. There is a recent advancements. Maybe it is coming to India also, but it is costly because where like the most of the cancers, even where the doctors cannot treat, in that cases also the cell culture or this gene therapy is used now. Like this CAR T cell therapy is nothing but chimeric antigen T cell receptors from the patient's blood. They'll take out this, they'll train by using genetic engineering the cells to target the cancer cells, and the cell can be grown in cell culture, and these grown cells will be introduced back to the patient. So this goes and target the cells. Like that, there are so many applications that are there for the cell culture technology. And this can be used for disease modeling, like to study cancer, genetic diseases, or infectious diseases, like metabolic disorders, and different things can be studied by using cell culture. Next. Yeah, these are the different terms I already told you. Maybe confluency. Maybe you can ask me, like, if you want whatever the term you want, I'll explain you. Yeah, passaging is the term, nothing but the generation, different batch, we can say. Like if when you take a cells from direct source, that is called first passage, for example. When you subculture from that, no, that will be passage 2. Like that, there are different passages. The passage number is very important in doing cell culture experiment. That is depending on the cell type. For primary cell lines, we can passage only up to 5 to 10 passages only. Other than that, the cells cannot be grown; they'll be died. Even for continuous cell culture also, so maybe up to 30% can be grown. There is a limit, but these cancer cells, when they got immortal, so the cells can be grown for multiple passages. But whatever the passage it is, so whenever you're doing experiment, you have to limit your passage number. So always when you're growing cells, you have to freeze more cells, and try to finish your experiment within 10 passages from the cells you started. So the medium with FBA, without FBS is plain media, and with FBS is complete medium. Subculturing bacteria, culture mixture, culture into pure culture. It's like subculturing bacteria. Yeah, it's not like culture means like I have cells in my flask, like the T25 flask can be grown 2 to 3 million cells. So subculturing nothing but splitting. Actually, we use the term splitting. Like there we use subculturing. So here for splitting nothing but when I grow a complete cells, cancer cells in a flask, so I want more cells so that I can trypsinize that cells. I can take out all these cells. I can split into different flasks so that I can that's like this culture can be added to more flasks so that we can grow more cells. It's like splitting. Subculture is nothing but. So just another one. Yeah. Any more questions?
Hello, sir. I'm Prerna.
Yeah.
Sir, actually, there is a one term you told ki you use trypsin for detachment, right?
Yes.
So, sir, trypsin is an enzyme.
Yes.
So,
Yes.
you told ki you use 0.25 something.
Yes.
Percent of
0.25%.
trypsin.
Yes.
Yeah.
Trypsin EDTA.
But, sir,
Yes.
yeah.
Yes.
But, sir, it is enzyme, no? So,
Yes.
if the trypsin concentration is more, it is the
Yes.
more chances to engulf the cell also.
Yes. Very good question. Actually, yes. Yes. It is not good for cell, but the trypsinase process goes only for 2 minutes maximum. Within that time, so when we add it, we'll just keep it in 37 degrees incubator, CO2 incubator. Within 1 to 2 minutes, the cells start detaching. So that we immediately will remove the trypsin, and otherwise we'll neutralize the trypsin by adding double the amount of trypsin. Like for example, T25 flask, we add 1 ml trypsin to detach the cells. To that, we'll add 2 ml of complete medium. So this media will neutralize the activity of the trypsin, so that the cells won't be affected. If you just keep it for longer time, more than 5 minutes, yeah, as you said, the cells will be affected definitely.
Yes, sir. Because, sir, I know ki there is a standard time like for incubator 3 minutes, no? So
Less than
you
3
have to
minutes.
keep it in.
Actually, it's like 1 to 2 minutes only. Less than 1 minute sometime. It is again depending on the type of cells.
Okay. But, sir, is there any possibility we can use this trypsin is a chilled trypsin like that when it is very cool and then we can use?
No. Whatever you use, like including media, FBS or PBS, whatever you use, everything should be in room temperature. Before working, we have to take them everything out. That everything should come to room temperature, because when you add, I mean, this media is directly from the refrigerator. That cold temperature will cause like discomfort to the cells. The cell may like behave that they may lose some their properties sometimes.
Yes,
So
it is in stress like.
yes, they may undergo stress also sometimes. That's what everything before 50. I mean, before you start working, we will be switching our UV for 15 to 20 minutes. When we enter into cell culture, generally we take out all the medias, whatever we are going to use it. So we'll take them out and we'll keep them outside. Then we'll switch off the UV in the flow as well as in the room. So that meanwhile, that 15 to 20 minutes, all the reagents will come to the room temperature. So we'll be adding. Even that cool when we.
And
Yes.
sir, if we are work on suspended cell lines,
Yes,
so
yes.
how to split this means
Yes.
we cannot use the trypsin and all.
Yes,
So we
yes.
have to do the centrifugation. But if
Yes,
we
yes.
centrifuge the cells, then it will be again is in stress also.
No, no,
So
no,
how
no,
to
no.
seeding?
Yeah, yeah. Yeah. Actually, working with suspension cells is very easy actually compared with the adherent cells. So for adherent cells, you need trypsinization again. Maybe when you neutralize, you have to centrifuge it to collect the cell pellet. So for suspension cell lines, generally the cells are dense, so they'll be pellet down in the bottom. So actually, when we keep the cells into the CO2 incubator, the suspended cells will be like in vertical position, the flask. Okay, and for this adherent cells will be keeping in horizontal. So like when you keep that, the cells will be like in the bottom mostly. So like when you are walking, so we can take out the supernatant, the cells in the bottom only without disturbing them. Even centrifuging up to 1500 RPM or 1500 RPM generally we use. Even up to 2000 RPM also you can be used, but mostly 1200 to 1500 RPM is suggested. So the cells won't get any damage even for that speed.
Okay.
Yeah.
Okay, sir. Thank you.
Yeah, I think there are some questions. Yeah, thawing process. Yeah, freezing. I think maybe at the end it is there. Maybe I'll discuss there. I'll tell you. Why only trypsin enzyme are using for breaking down cell extracellular matrix? There are other components also. Yeah, there are some other, but we generally use this trypsin. 0.25% we will be using. This is commonly used. Maybe we can use some other enzyme, but maybe this is completely studied. Maybe we are using. Yeah. So next slide. Yeah, so coming to the types. So by the source or duration, primary cell culture. So the cells isolate from directly source from any tissue or organ when you grow it in cell culture, that is called primary cell culture. So like they have very limited lifespan. Maybe I told you like different limited, like up to 5 to 10 passages only you can grow them. And then another type is secondary cell culture, where maybe the primary cell culture subculture or primary culture into a new vessel often necessary the growth after primary cell culture. This cover the surface area, that is secondary culture. So by the growth characteristics you can see adherent, I told you already, adherent are monolayer cells attached to the surface. Example, you can see HeLa, which is a cervical cancer. MCF7 is a breast cancer. FG2 is the liver cancer. And coming to the suspension which like float in the media, that is example THP1, which is a monocyte, and HL60 and K560, these are a leukemia. Both are cancer cells only. Yeah, next slide please. So you can see different cell line, finite cell lines and continuous. So cells can only divide for a limited number of time before dying, often derived from a primary cell culture is finite. And continuous we can say like immortalized cells which have undergone transformation due to mutations or something, genes. So they can divide indefinitely, so we can grow them in more passages. So again, based on the morphology, you can see there are cells are mostly this epithelial, lymphoblast and fibroblast type cells can be seen in cell culture. So you can see epithelial like attached. Maybe next slide. Can you just move to next slide, sir? Yeah, here you can see epithelial like cells, fibroblast like cells, and lymphoblast. These first two are adherent, and here you can see the lymphoblast is suspension. So the cells based on the morphology, you can see these are the three common types which we see in regular cell culture. Yeah, next slide. So you can see some examples. These are the human cancer cell, and you can see MCF7. I told you breast cancer. And here one more thing: when you're doing, when you're screening some compound for anti-cancer activity, you have to test on two same type of cancers. For example, you are working with breast cancer, MCF7, MDMB231, both are breast cancer, to different origins, and with one control cell line also we have to use. So then only the data is valid. Or sometimes you can use one single cell line. It depends. Some journals will ask you for 2 cell line, 3 cell lines according to the journals. So they will be repeating again. So HL60, which is leukemia, HEK293, which is a normal cell line, which is normal. It is not cancerous cell line. Human embryonic kidney cell line is very commonly used cell line for as a control. And HeLa is a cervical cancer. And along with that, HeLa, CHA is there, which is also cervical cancer. So you can see primitive cell lines also, Vero, COS7, and here you can see CHO. I mean, this is from hamster. Next, sir. So like you can see the process simply from any source, you can isolate an organ. So that organ culture, like any kidney, can be taken from tissues into the, just you can cut it into different pieces. If you grow it primary cell culture, and when you just passage it more and more, that will become a cell line. So we can take any like fish, and generally we can use some liver, kidney. Such cells can be grown actually easily by using suitable mediums. Yeah, next slide. Yeah, here. So coming to this, so we know that now we know the cell culture, their applications and the types. So what are the basic requirements which you need for maintaining cell culture? First one is laminar hood, which is known as class 2 or class 1. Anything we can be used laminar flow or biseptic cabinet bay, which is needed for growing cells or working with the cell lines. So like this all, you know, like they have filters. So the air is filtered. There are different classes depend on the requirement. Maybe class 3 is highly like when you are working with viruses only we need, but generally class 1 can be used for cell culture. Class 1 or 2 can be used even. There is a question again. How difficult is to develop a new cell line for research purpose? How to select cell line? Actually, mostly we don't develop, and maybe that will be like there is NCCS Pune is there. So generally we order from there because it's compared to other cell like ATCC and other things, they cost around 50 to 60,000. But NCC Pune will get around 5,000 cell line for government organization, 4,000 plus GST. So generally we can get the cell line like according to the literature, maybe your compound. So we choose cell line like our compound may be effective against cervical breast cancer. Accordingly, we can get the cell lines. For developing, no, it will take long time actually. For primary cells we used actually. For developing new cell and the confirmation, this will take long time. Maybe this NCCS and other things, these people will be doing regularly onto this. Okay, this another one is important thing we required for cell culture is along with the flow. Maybe flow can be used any flow, even normal microbiology flow. Like when you have a new flow that can be used when you are experienced, when you have full hands experience, it can be normal laminar flow can be also used when you are experienced only. And along with the flow, we need the major requirement of cell culture is CO2 incubator. So we can see here this incubator, no, so you can see there are different slats, and below you can see the steel container, which is like there we pour sterile water to maintain the humidity. So this incubator will maintain 5% CO2 continuously and 37 degrees temperature. So that is our physiological conditions are maintained in this incubator. So always we have to maintain the humidity, and there is a sodium bicarbonate system inside the medium. We'll use sodium bicarbonate and the medium. So the CO2 is very, this incubator is essential. Without this, you cannot work. Maybe normal flow you can be used, but the CO2 incubator is essential. Always 5% CO2 has to be supplied to the cells. Yeah, next. So the another important thing is inverted microscope. Generally, we have our microscopes for bacteriology that cannot be used for cell culture because the cells are mostly in the bottom. The cells are in the bottom only, so the object lens in the bottom, so we can focus the cells. So we need definitely without inverted microscope we cannot see the cells. So like different lens, maybe 4x objective also we can see the cells. 4, 10, mostly we use 4 and 10 regularly. Maybe for imaging, sometimes depending on the requirement, live cell imaging also we can be used. So this is a basic inverted microscope. Yeah, next slide. Yeah, as I discussed, there are culture media. So like there are different medias which contains salts, carbohydrates, vitamin, amino acids, metabolic precursors, growth factors, hormone, other elements. So which are there in the media. So the media contains all these elements. So there are different, I told you, commonly used culture media, that is Eagles medium, minimal essential medium, Delbecque's modified Eagles medium, and RPMI. There are different medias. So I told you now, like earlier the medium was so cost. Now I think there are different companies also started preparing the media. Now we are getting it at around 1,500 to 2,000 for 500 ml of any medium, plain medium. So earlier we used to get powder. So that powder will be for around 2 to 3,000. We can prepare around 10-liter media with the powder. But again, you need to purchase this antibiotic, antimycotic solution, syringe filters, syringe, and you have to be like take care, like unlike the commercially available medias. So you have to be prepared always regularly whenever you need. Like we cannot prepare 10 liters media at a time, and we can store whenever we need. like for example, I just used to prepare 2 to 3 days medium, which is required sufficient for 2 to 3 days, like 500 ml, 200 ml, 300 ml, like that. So we used to prepare and we can store it in the 4 degrees temperatures. Yeah, next one. So here I told you already, complete media and plain media. You can see the middle one is yellow color is ABS. This last one is trypsin EDTA 0.25%. And here you can see the different media bottles. Yeah, next one. Yeah, here you can see. Yeah, we need flow, CO2 incubator, microscope, and we need even centrifuge also. And Newbar chamber is required, and centrifuge, and minimum 4 degrees to store medias or whatever the things, and -20 to store like ABS and other things can be stored in -20. Like we can like take limited amount of things. Working stocks can be maintained in 4 degrees. So along with that, there are different culture plates, different media, different things we can be used in cell culture. Here you can see, I think this flask, no, blue colored cap one, these are tea flask. You can see tea neck. It's like tea neck. That's what we call it as tea flask. So you can see the bottom surface area. That is the first one is T25. So there is a 25 centimeter area, and below that there is a T75 centimeter. Maybe you can see the like petri dishes, like generally we use in microbiology. These are cell culture treated petri dishes. Even we can use different size of petri dishes for growing cells. And you can see 96 well plate, 6 well plate, 24 well plate, like that. There are different plates that is according to the requirement or the experiment you are doing. So for example, if I'm doing any cytotoxicity testing, I'll be using 96 well plate, like that. When I'm using some protein work, I'll be using 6 well plate, or sometimes we'll be using T25 flask directly. It depends on how many cells you are using. Like T25 flask, I told you 3 million cells can be grown up to 3 to 4 millions, like that. Yeah, there is a question. Yeah, just previous slide, sir, once. Yeah, I want to tell you one more thing. You can see these caps actually. There are flask are 2 types, vented and plug seal. Vented means this flask has holes, that filter like types. The air which goes inside is filter. It is trapped. Any particles will be trapped. Another type is plug seal. So like for example, I order cells from NCCS Pune. So they has to send cells from Pune to Hyderabad. So how the cells can be like transported? If we are using plug seal, there is a like ventilation so that the media can be leaked or that can be get contamination. So they use plug seal. Some type of cells can be used like complete airtight. So when you are using plug seal flask, we just loosely close the cap. When we are using vented flask, we can close it tightly because there is a ventilation for it. So there is no contamination. So there is no chance of contaminations. The lid is closed only for vented. Even for plug seal, the lid is little open, so there won't be any contamination. No problem. Okay, next one. Yeah, so here there are different things. Just you can see.
Sir, actually,
yeah. Yes.
sorry to interrupt, sir.
Sorry.
But sir, you told there is 2 flasks, T25 and T75.
Yes,
So
yes.
sir, I saw that type of flask. They have different color in that flask,
Yes.
like cap color. One
Yeah,
is white, one is blue. So what is the purpose of that color of cap? Means
no, there is no purpose actually. The color is like different companies. Like Talson will be in blue color. Maybe some other biofuel companies in green color. There's nothing actually. Only we have to
it
look.
means
When
nothing is coated on that tube.
No, there is nothing related to the color in the flask. Only we have to check whether it is a vented or plug seal. Maybe you can see there is no white wheel-like structure in this flask. Maybe you can see right side of this. This flask, there is 4 flask right side. So you can see that white color. You can see something. This is ventilation. This is vented flask. There is nothing with the color here. So these are different filters you can see. Like when you filter media, like we cannot sterilize. No, I told you, I think. So we use filter sterilization. We use 0.2 micron filters, membrane filters to filter the media, to sterilize the media, because we cannot autoclave the medias. So these are different plastic where we can use 50 ml flask. Yeah, one more thing I forgot to mention. Like some cell lines, like RAM macrophages. So those are adherent cells only, but the cells, when you add trypsin to the cells will transform to some other cell. Sorry. So such cells we cannot use trypsin. In such cases, we use scrapers. Maybe in this slide, you can see this green colored, these things, no, with white ends. You can see beside the ependraft. These are scrapers, cell scrapers. We use scrapers, such cells. Or maybe trypsin is not available also, you can scrape the cells. Gently, we have to scrape the cells. We can collect the cells, no problem. So these are different things can be used.
Okay, sir.
Yeah, next slide, please. Yeah, so the major risk, major problem with cell culture is contamination. So that may be with chemicals or biological agents. So there are some toxins, other things can be interfere with the cell culture work, and that leads to contamination. So the biological agents mostly will get mycoplasma contamination, sometimes fungal contamination. Mostly we use fungal and mycoplasma. This mycoplasma contamination cannot be seen easily because that can enter into the cells, and that's what when we were growing the cells or when we want to do some experiment, we'll grow the cells for 2 to 3 passages first. Then after third passage only we'll use them for the cell, our experiment, because to see the morphology, is there any differences, any variations, any contamination. So first we'll grow the cells 2 to 3 passages. After third passage only we'll use the cells for the any experiments. So that is important to like find out, to rule out the contamination before wasting your compound. Sometimes you may have limited compounds, like even the medias are limited sometimes. So to avoid all these things, it is better to maintain, grow the cells in 2 to 3 passages. Then if the cells are well, their morphology is good, then only we'll proceed further for the experimentation. So like there are different, these contaminants can compete with the nutrients with the host cells, and even they secrete different compound that leads to these things, and they degrade other, yeah, and they produce some peroxides, other things that can be toxic to the cells. Yeah, next slide.
Sir, I want to ask you a basic question about this. Actually,
Yeah, yeah.
you said primary cell culture. So I am a little bit confused about how is it primary cell culture after 3 to 7 it gets cell line or we, because when we started primary cell culture, it is culture of mixed population of cells. Suppose so how we get this particular cell type cell line from that primary cell line?
For example, you want to grow some liver cells, you can get liver, you can just make, mince it and you can grow it. So only one type of cells can be grown. That can be like when you are taking from the source, directly from the source, that is primary cell.
Yes, so
Yeah.
like primary cell contains some other cells, like in the microbiology, like we
Yeah.
take soil sample, it's mixed population, then we subculture continuously so that we
Yeah.
get the pure culture of single type of cellular colony.
Yeah,
It is like
yeah,
this.
yeah. Here, like no, so like we are taking, say, only one organ. Like for example, I want to grow liver cells only. I'll take only liver. Other cells may be there, but the majority is the liver. Like maybe other cells may interfere, like blood cells will be there. But the cells, no, these blood cells and other cells are floating. They cannot attach. So only the liver cells can be attached to the bottom and will be continuously washing. Will be seeing for the morphology. Then only, and we are not establishing the cells, but doing some basic experiment will be growing with a primary cell line. Mostly we'll use continuous cell lines only, secondary cell lines only, or most of the cell culture will be used, not primary cells. But primary cells, like some cases, no, when they want, like when it is not available, we can grow them.
Okay, so we should create primary cell line, then secondary cell line, then we shift it to cell line.
Yes. Secondary is no, like it when it is like established, after growing some certain passages. So that can be like after passages only we can be, that can be like we can consider secondary cells. When, yeah, as you said, like when we purify, when you like purify and characterize, then only that can be secondary.
Okay,
Okay.
it's like
Yeah.
microbiology, we follow the colony morphology. If we follow in microscope
Yeah,
examination.
yes. Here also we can see under microscope, you can see the cells direct, we can see the morphology, and we can see any like contaminants or other cells are there also we can see. But like in microbiology, you can pick a single colony, we can grow it in subculture and we can do that. But here it is not like that. So maybe like we'll be using different enzymes to degrade other cells, and when you want to purify the cells, but we are not into that level. We don't do such things. We'll be using already ready-made continuous cell culture cells. We'll get it, we'll order, or we'll borrow from IICT. We have nearby CCMB, NIN and all. So we'll get, we have our friends, no, most, if it is not available, we'll order from NCCS only directly. So it is already characterized, So studied cells only will get it from there.
Okay, sir. Please.
Yeah. So here you can see the contaminants. Maybe generally you saw the media which is in red or pinkish color, but like when they get contamination, you can see they will become turbid like this. You can see there are different color change will be there because like you can see the complete like the medium is clear actually. The cell culture medium is very clear and you can see them. But when they get any contamination, so they start showing the turbidity is a major sign. And then you can see this under microscope, you can see the picture. The cells will like that. Their morphology, like they lose their morphology. They can see like internal things like this. So like when you compare with the normal cell, the morphology completely degrades. So likewise, we'll get the contaminants, and mostly when we get any contamination like this, very severity is there. So this happens sometimes when you are working with multiple cell lines at a time. Like even one cell grown in some other cell line also, that may be a contaminant. Okay, so such cases also that leads to contamination. Pipettes, if you are not changing pipettes, any change like even the flask, if you are not using it properly, so that leads to contamination. So once we get the contamination, mostly we try it because we'll have a lot of backup. Flask cryo wells in our this thing. So mostly we'll discard it and we'll fumigate the lab and we'll start it again. But like sometimes only when it is started, when it is in initial stage, when the like you have the cells, maybe it is not available, we can give antibiotic treatment and we can revive the cells. It will take long time actually. It may take 1 week to 10 days, so that even we can slowly we can revive the cells. When we can get rid of contaminations when it is initial state. When it is completely the morphology is, yeah. Yes.
But sir, I read about it. Means I have just theoretical knowledge about that.
Okay.
So sir, I read ki the media component have some component like phenol red for pH indicator.
Yes.
So if we grow the cells on that, so it will change automatically media color.
Yes. Actually, when I'll come to it. So when the cells are growing, definitely they'll release some toxins, the metabolites. Even that leads to change of media color. That
Yeah.
media is clear only, but the color is changed. It is clear. There is no
Okay.
turbidity. There is no turbidity. There is a difference in growth of cells, metabolites, and other things. We can differentiate it. So, but when you get contamination, the media completely become turbid. But when the cells are grown, that the media color change, mostly in yellow color, they'll become acidic.
Okay.
So that means you have to change the media. That means, so the cells are grown. That is an indication actually. It's not a contamination. Yeah. So there is a difference
at the time
between
of passaging, we have to just change the media.
Yes. Because for example, when I just seed the cells, so some cells will grow very slowly. For example, MCF7 cells will take 2 to 3 days to get full confluency. So when generally in T25 we'll add 5 ml media, 5 to 7 ml media. So in single first time I added 5 ml media, that may not be sufficient for 2 to 3 days. So regularly we have to change the media. We have to take the old media and we have to add the fresh media. So as the cells growing, they'll release some metabolites, right? Yeah. So that we have to, yeah. One more thing, when you're working with cell culture, you have to be very regular, like it is Sunday or anything, any festival. Every morning and every evening, 2 times at least, you have to visit your cell culture. You have to check your CO2 levels and that humidity, water, and even the media color is changing, how the cells morphology. We have to check it regularly. So accordingly,
And
you have to
after 2 days, we have to revive also, no?
Yeah, for example, the media, like if it is in color, red or pinkish color, it is okay sufficiently. And when it is changing, so we can change the medium.
Okay.
Because the cells are attached to the bottom. When you take out the medium, the cells won't come out even. So we can take out the spent medium, we can add the fresh medium, no problem. That can be done.
Okay.
That is until you get full confluency, maybe 2 days, 3 days sometimes. If it is T75, it may take 4 days, 5 days, like that. And that is again depending on the cell type and depending on the culture, how much culture you added. That splitting ratio. Sometimes 1 flask can be split into 2. Sometimes it can be split into 4. So when you split into 2, within 1 day, you may get full confluency. When you split into 4, that may take 2 to 3 days, like that.
Okay.
Yeah.
Okay.
Yeah. Next slide, please. Yeah. So, yeah. Then coming to cryopreservation, so like I have some experiments, so I got the cells from somewhere, maybe I purchased. After my work, and even during the work also, we were continuously growing the cells. So because we cannot order always, because when you order, that will take minimum 2 to 4 weeks to reach, so that you have to waste a lot of your time. That's what when we get the cells, we grow them in many we can freeze them in the we will do it cryopreservation. So here for cryopreservation, we use cryo vials, as you know already. So here we use DMSO as a cryoprotectant, like as we use glycerol for storing bacteria. For cancer cells, we use DMSO as a cryoprotectant. So you know the role of cryoprotectant here. Even again the question, like even the DMSO is toxic to cells again. So when you add the DMSO to the cancer cells or any cells, the cells start maybe dying, or maybe that get maybe into stress. So as when you add it immediately, will be shifted to the -20 immediately. Then we'll keep there for 1 to 2 hours. Then from there we'll shift it to the -80. Then again from there we'll shift it. If you have availability of liquid nitrogen, will be shifted to long-term storage. We'll store it in the -196. So whenever we want, we simply thaw it. That by using water bath, 37 degrees water bath, we thaw it. And again there is a question. So when you thaw the cells, again the DMSO will get activated. So to neutralize the DMSO, we use 10% DMSO for freezing the cells. So like generally that vial no cryovial will be like, we'll add 1 to 2 ml of medium, depending on the number of cells. If it is 1 million, 1.5 million, 2 million cells, so that vial will be transferred to the 15 ml tube containing around 15 ml media, so that that percentage will be decreased. Then immediately we'll centrifuge and we'll take out the supernet, and then the cell pellet will be resuspended in the fresh media. And likewise, just will revive the cells. Next, sir. Yeah. So yeah, coming to some basic conditions. So yeah, always like we have swapped all the bottles, not only bottles, whatever the things. Maybe it may be like I told you, like scraper, syringe filters, syringes, media bottles, tips in bottles, whatever you take into the flow, we have to wipe it with the spirit or ethanol. We use 70%, 90% ethanol will be used. And so the pipette and when you are using serological pipette, we have to be very careful. And we have to place the caps always down on the bench, not in the upright positions. And when you are working on the cell culture, you are not supposed to take your hands above your media or culture plates, because something which is there to your hands maybe fall into that leads to contamination again. So we have to be very, very careful while doing this work. And you have to work either left to right or right to left, like that, vice versa. And all metal goes to one side and finish to the other side, to like remove the tension while working, maybe this thing. And you have to clean up the spills immediately. Always leave the workplace neat, like before and after work. You have to sterilize your laminar hood clearly with the alcohol. The next one. So yeah, you have to keep the cultures free of antibiotics in order to be able to recognize the contamination. Sometimes medium, like whenever we want to test it, when we get contamination, we'll rule out whether the contamination is with the media or FBS or with PBS or the whatever, where is the source, we'll try to find out it. So for that, we just keep only plain media into the flask and we'll keep it in the incubator. We'll check it for the contamination, like that. And we don't use media bottle. Same for different cell lines. Yeah, here one more I forgot to mention. There are different medias, like bacterial media also. There are hundreds of medias. Like, so even cells also, we need different types of medias. Like for example, I want to tell you one more term that is called media adaptation. Okay. So for example, I'm working with some HeLa cervical cancer cell line. So I'm using DMEM media. I have sufficient media now. But like I just when there is a DMEM is a shortage, so I have a lot of another media like MEM, for example. So like I cannot use the another medium directly to the cell lines, because yeah, one more thing we have to mention clearly is when you are working with the flask, I show you this T flask, you have to mention the type of cell line and passage number always in the flask, passage number and the medium used and the date we have to mention clearly. So that, for example, when I am working, some other people also may enter to know to some, maybe I may not available. I may ask my friend or lab mate to change the medium or to split the cells. Such cases, he cannot ask me all the things. So that information should be there in the flask, everything, so that accordingly the person can work on it. So we have to maintain all these things: type of cell line, passage number, and medium used always, so that the same medium can be used. If you, instead of DMEM, if you use MEM, the cells may not grow, because that will take time again. But in such cases, when you don't have that medium we are using for cell culture, when you are growing cells, you have another media, so you can adapt the media. So cells can be adapted to the different media. Like you can use the media whatever you are using, 90% plus 10% of media which you are going to adapt. So 90 is to 10, 80 is to 20, 70 is to 30, like that. Slowly you can adapt the cells into different medias. This can be done. Another thing. Okay, and the glass bottles prefer to eat that heating we don't use mostly. Sometimes the caps and other things can be just show to the flame. And the laminar flow, as you know, microbiologist. So I feel like microbiology people can work. I mean, we can learn cell culture very easily. I learned within 2 weeks actually complete cell culture. During COVID time, I started with my senior. Within 1 to 2 weeks, I started doing my own, maintaining the cells, splitting everything. So like as we know microbiology, like how to do the things, it is very easy to handle with the cell culture, easy. But the things are different, the terms are different, and we have to be more careful that simply. And before and after working, we have to switch on the laminar flow, and so we have to be like freeze more and more amount of cells, because we cannot purchase cells always. That is another thing. I think there is a question. We mostly used -20 for long-term preservation. Is apply here also, or done only cryopreservation? Yeah, long time we can store it in months to years also, but it is preferred like when you are storing more than months, 1 or 2 to 3 months, it is better to store it in liquid nitrogen. And the serum-based media also, I didn't get the question. Yeah, even while freezing, generally we use 10% ABS, but freezing we use different percentages, like 20%, 30, even some use 50%. If more the ABS, so that will cushion the cells, and the cells cannot be damaged. Yeah, next slide. Yeah, is any serum based media for? Yeah, definitely serum is it's essential. Without serum, the cells cannot be replicated. We'll be using 10% serum always. Only sometimes we use serum free media, that is to make serum starvation. I told you the term serum starvation. Only such cases we don't add a serum. Otherwise we use ABS, commonly used 10% ABS will be used always. Then only the cells can be replicated. No, for storing cells without serum we cannot. The cells cannot be stored. They'll undergo stress, they'll die even. So definitely minimum 10%, maximum up to 50. This depends on your like regularly how do you do. Generally I use 30%, 20 to 50% used. It depends on the cell types. I use 20%, 30% ABS for storing cells. Okay, so you can see always I told you actively growing cells, like as we activate the cells before any experiment like bacteria, here also we have to when the cells are frozen, we have to thaw them, we have to revive them, we have to grow them, and we have to use them, and always 80 to 90% viability. Yeah, one more thing, we have to check for viability always when you are thawing the cells, when you are growing the cells, when you are seeding the cells. How to check for viability? How do you know whether the cells are viable or not? So one is by microscopic observation. As you regularly work with a certain type of cells, we know the morphology and how they look. And another thing is trypan blue is the dye which we use to compare or to know the viability of the cells. So generally, trypan blue is we use to differentiate between the living and dead cells. So when the cells are above 50% only, then only they can be seeded, even can be revived. If the cells are less than 50% active, no, so we don't use it generally. So then yeah, I told you now, as you already asked the question, keep the exposure to trypsin is a minimum. It's less than 2 minutes mostly preferred, sometimes even 1 minute, even sometimes 30 seconds they start detaching. And hardly cells gently, and do not centrifuge cells at height speed. Generally I use 1200 to 1500 only, not more than that. Even some people use 1800, 2000 RPM also, but I prefer, it is depending on your own experiences. So generally I prefer 1200 to 1500 only. And feeding subculturing the cells at more frequent intervals than used with serum containing condition may be necessary. So like maybe you may having 2 to 3 days holidays sometimes, so you may not visiting the lab, so you may add more media. The people will think I'll add more media so that will be enough for 2 to 3 days. It's not like that. So we have to be keep on regularly, we have to watch. Maybe one day we can manage sometimes, but we have to be very regular, and we have to continuously regularly change the media according to their behavior, like the change of color of media, their morphology and all. We have to consider all these things. Yeah, next slide please. Yeah, this like I covered most of the things. Like if you have any questions also, you may like you can ask me or you can post it here and you can come to my contact or mail ID. So here she's my professor, Dr. Professor Anita Burgla. She's my PhD guide. Like earlier director CFRD Central Facilities for Research and Development, Osmania University. So I always thankful to my madam because whatever the knowledge I gained, that is from my madam and my senior. So I'm always thankful to madam because along with madam, we have conducted so many workshops, training programs in the university. I think more than thousands of people were trained under madam. So I'm fortunate to work under madam so that because of her only, I got all this knowledge because I'm sharing with you all. So I'm thankful to madam. Yeah. Next. So this is about my company actually. We were providing different like things to the students, like academic trainings, projects for BSc, MSc, other all life sciences. We will be doing some trainings for students and other things which are located in Hyderabad. Actually, I'm from rural background. Up to BSc, I'm pure Telugu medium student. Only MSc English medium. So I studied everything in government institutions only so far, if up to now, even for PhD also. So I struggle a lot to reach this stage. So I don't want any student to struggle like me. There is a gap between industries and students. The students lack a lot of hands-on trainings because of which they are not unable to get the positions, placements. So to fill this gap, I started this lab to train the students in real hands-on experiences. I want to help the students where I lack during my studies. I want to help the students. So this is the motto for establishing my company. So I'm trying to do this and filling the gap between the industry and student academic. Yeah. So next slide. Can you go to next slide, sir? Next one. Next. Yeah. This is just to show you some activity which is done in my lab, PA's lab. So there is a like Andrographis paniculatus. There's a plant extract we have tested for. Here you can see this is MTT assay. This top one you can see blotting, no? So even blotting like protein expression also can be studied by using cell culture. The second graphs, different colored graphs is, no, this is MTT assay, cytotoxicity testing. And here you can also analyze the cell cycle arrest. You can do flow cytometry analysis for cell culture by using any compound, like where your compound is arresting the cells at what stage of cell cycle. And the down one, you can see some scratch, some road-like things. No, this is called scratch assay, also known as wound healing assay. We'll be regularly doing to know the compound, any anti-cancer wound healing properties of your compound. And here you can see the CAM assay, you can see the blood vessels. So these are the common assays we'll be doing regularly in cell culture. Yeah. So I think I cover maybe 1 2 hours may not be sufficient to cover everything. Maybe I tried to cover everything. Maybe I may miss some things somewhere. Maybe you can ask me, you can contact me like through any mail or any through mobile. So like any questions can be asked. Yeah, IC50, yeah, PPT, yeah, will be shared to sir. So IC50 value, no. So yeah, so like we'll do no MIC for bacterial this analysis. Here also we'll be using IC50 values also can be calculated by using this assays, cell culture assays. Yeah. Yeah. Any more questions?
If anybody have any question, please ask to the sir.
You impart training also for the students?
Yes, sir. Yes, definitely.
How much you charge?
Like it depends. Actually, we'll be conducting regularly through OU, but now I'm out of university. But I am collaborating with university to conduct these sessions. So this is like it's for 1 or 2 days program. We'll be charging 2,000. It depends whether it's a demonstration session or hands-on session. So like from my company now we are demonstrating, like we are taking cells and this microscope, inverter microscope to the institute, some colleges, because we cannot get all the students to our institute because there is limited the spaces. So sometimes according to the requirement of the some institute in Hyderabad, so we are going there and I'll take the class and we'll demonstrate like how to split the cells, how to trypsinize, how to count the cells, how the cells looks like really, and different things, whatever we can do it in 1 or 2 days programs are conducted. But if anyone want like complete hands-on training, that may be like 1 week or 10 days. So like the fees are like not yet finalized. Maybe it will be like around 5 to 10,000 in between, depends on the duration and what all techniques you want to learn.
And how much you charge for analyzing the samples? Or that you have to start it?
Yeah, this is like MTT assay is the basic cytotoxicity assay. It is around 1500 to 2000 for each cell line per compound.
Mhm.
Yeah. So like 4 to 5 concentration along with the positive control drugs will be testing, and will giving the methodology and everything. All the data with analysis will be sending, like whatever the requirements. It's for basic cytotoxic assays, and we'll be doing CAM assays also.
So that you do in Usmania or
Yeah,
in your
earlier I
company?
used a Usmania. Now we are establishing at my company also. I have access to some 2 to 3 private labs also. Within 15
Very
20
good.
days we are going to establish completely. Yeah.
And then cell line you are getting from NCCS Pune only.
Yeah, we mostly like whatever we don't have, we'll order from LCA. Because
Hello.
yes, sir. Hello.
Hello, Ramesh.
Yes, sir.
We will conclude now. It's more
Yeah.
than one
Yeah.
and a half hour.
Yeah. Okay, sir.
So, Asmita, proceed.
Yes, sir. So, good evening, everyone. On behalf of the Microbiologist Society, I would like to express my sincere gratitude to our esteemed speaker, Dr. Ramesh Kande, for delivering such an insightful and informative session on cell culture techniques and its application. Sir, your knowledge and practical insights have truly added great values to all of us. I would also like to thank Dr. Arvind Deshmukh, sir, for his constant guidance and support. My heartfelt thanks to the entire organizing team for their efforts in making this session a success. And finally, thank you to all the participants for your active presence and engagement. Thank you, everyone. Have a wonderful evening.
Yeah. Thank you, madam, and thank you, sir. Maybe you can share my contact details so that they can contact me for any doubts or any sessions. I'm always ready to answer. Thank you, sir. Thank you so much. Thank you,
Yes, sir.
Ashmita madam, Deshmukh sir.
Ramesh, sir.
Yeah, ma'am.
Sir, I'm excited to learn these all things like flow cytometry and all. So I have requested to Arvind sir, please can you arrange again Ramesh sir lecture for flow cytometry?
Flow cytometry, like we don't do it directly because we don't have that facility. We'll give the cells. We'll treat all these procedures. We just give the cells for like other facility wherever like NIN and CCMB.
Okay, sir.
Yeah,
Sure.
only the cell culture part, all these things we can be done. The cell culture, flow cytometry, we don't have the facility. Maybe
Okay.
regarding cell culture, we can, like, if you want any specific assays like CAMAS, if you want, I can come and demonstrate if it is like I'm ready to come anywhere. Even for these sessions also directly. Maybe if you have facility, if you want to establish the facility also, I can help you to establish anywhere.
Okay, sir. Thank you very much. Yes, sir. Thanks.
Yeah. Any more questions? Yeah, for certificate, you maybe I don't know. You have to ask, sir. Yeah, thank you everyone for like attending this and patiently listening to my lecture for last 1.5 hours. So I'm always ready to, like, if you need anything, you can just get into contact. So we'll be discussing. If you have any experiment also, I can help. Okay, thank you, ma'am.
Okay, sir. Can we end the
Thank you so
session?
much, sir.
Sorry.
Thank you so much, sir.
Yeah,
I'm
thank
Jyoti from MIT University, Rajasthan.
Yeah, okay, thank you. Yeah, you can take my contact. So you can search me Ramesh Kande and LinkedIn, and anywhere you can get my contact details, or you can search through Micro Life Sciences also.